Green sulfur bacteria harvest light through a highly structured photosynthetic apparatus. Photons are captured at the base plate and must eventually reach the reaction center to be converted into chemical energy. To cross the gap between these components, excitonic energy moves rapidly through a biological conduit. This conduit is the Fenner-Matthews-Olsen, or FMO complex. It acts as a molecular wire. But energy transfer within this wire isn't always a complete circuit. The flow of excitonic energy through the FMO complex is actively gated. The structure can shut down the transfer entirely. Zooming into the atomic structure of the FMO complex reveals the mechanism behind this sudden stop, a specific amino acid called cysteine. Cysteine contains a highly reactive thiol side chain, a sulfur atom bonded to a hydrogen atom. The chemical state of this thiol group acts as the physical switch. This reactivity presents a severe structural challenge for chemists. The volatile property making cysteine an effective biological switch makes it difficult to manipulate in laboratories. To study or synthesize this molecular wire, chemists have to deploy precise masking techniques. They must temporarily silence the reactivity they eventually want to harness. Establishing control over these thiol interactions is necessary to transition from observing natural photosynthesis to engineering synthetic light harvesting systems. This is a 3D model of the FMO complex. Deep within its architecture are two highly conserved cysteine residues, Cis-49 and Cis-353. Under normal conditions, these residues are in a reduced state, retaining their electrons. When reduced, the gate is open. The surrounding protein structure and electrical environment remain stable, allowing excitonic energy to flow unimpeded from the base plate to the reaction center. However, when exposed to environmental stress or high-intensity light, oxygen levels fluctuate. These conditions cause the cysteine residues to oxidize, losing their electrons. This oxidation causes a structural shift. The energy flow is quenched and safely dissipates. This sudden quenching is a biological necessity. It protects the fragile reaction center downstream from oxidative damage during a surge of light. By shifting between these two redox states, the FMO complex functions as a biological switch, tethering energy transfer to the chemical environment of the cell. Replicating this precise mechanism means transitioning from the living organism to the laboratory, to artificially build the FMO sequence from scratch. The standard method for constructing these protein chains is solid-phase peptide synthesis, or SPPS. Chemists build the molecules step by step, linking one amino acid at a time. But introducing raw, unprotected cysteine into an SPPS sequence derails the process. Because the thiol group is hyperreactive, it attacks other parts of the growing peptide chain. It forms erratic disulfide crosslinks, tying the sequence into knots and halting forward growth. The reactivity that gives cysteine its evolutionary advantage acts as a roadblock for synthetic chemists. Proceeding requires a structural intervention. The solution is a strategy called orthogonal protection. Chemists mask different reactive parts of the molecule with distinct chemical shields that only respond to specific, separate triggers. To synthesize sequences like the FMO complex, researchers use a derivative, FMOC cis-trit-OH. This molecule separates the protection into two distinct zones. First, the FMOC group blocks the N-terminus, ensuring only one new amino acid attaches at a time. Second, the bulky trityl or trit group is attached directly to the volatile thiol side chain. Its physical size isolates the sulfur atom, preventing it from reacting. With these shields in place, the elongation cycle of SPPS can begin. The first step in adding a new link is the targeted removal of the FMOC shield. Chemists introduced piperidine, a mild base. This basic wash dissolves the FMOC group, freeing up the N-terminus so the next amino acid can snap cleanly into the sequence. Crucially, the trityl group is unaffected by the mild base. Throughout the elongation cycle, the thiol remains safely neutralized. This cycle repeats until the FMOC sequence is assembled. At this stage, the protein is inert. The trityl group must be removed to restore the wire's function. To strip it away, chemists apply an acid cocktail, specifically trifluoroacetic acid. The acid dissolves the shield, exposing the raw sulfur-hydrogen bond of the thiol. By utilizing opposing chemical environments, a mild base to dictate chain growth, and a strong acid for final unmasking, chemists achieve precise control over cysteine's nature. We rely on heavy chemical suppression to build a molecule whose biological purpose requires rapid, unguarded reactivity. As the acid wash strips away the final protective tags, the synthetic chain regains the reactivity required for excitonic transfer. Successfully yielding this mature protein segment produces a functional molecular wire, capable of executing precise, redox-gated energy transfers. These synthesis techniques are required far beyond the FMO complex. They are necessary to construct the cysteine synthase networks that drive efficient photosynthesis in plants. Manipulating these sequences amino acid by amino acid allows researchers to map out and test artificial light harvesting architectures. Controlling the reactivity of cysteine allows researchers to precisely replicate the quenching and gating mechanisms found in the FMO complex and broader photosynthetic networks.